can
well, we are
accepting
on time.
the, yeah, I will admit
Yes,
the people.
I think we are on time.
You're gonna admit the people?
I will leave at 12 because I have to teach. So I hope we will finish the lecture, but even we don't finish, I have to leave because I have to teach in the university today.
Sounds familiar. Okay.
Yeah. Yes.
Okay.
So before we are admitting people to participate in the lecture, and while they are entering to the room, I will ask you to join, including Mona, to participate in this. I put in the chat a link, so you can complete the link to see where are all the people from. Oh, but it's not working. So you can click on the link, please, everyone.
Okay.
And put the city and the country where are you listening the lecture. So I will share with you at the end of the conference, or maybe now, so we can see.
We send by chat?
Yes, I send by chat. It's a link that is menti.com.
And how do I leave how do I go back?
You go back to Zoom.
I can't go back.
You should go to Zoom. We are looking to you, so you are not unconnected.
Here you are. Here I got it.
I already sent.
Good.
Okay, so I guess start, and the people will keep on entering. I guess no, we can maybe start.
Yes, yes. You can introduce Mona, and I will accept the people.
Okay, perfect. So for respect of the people that are already here on time, I would like to say good morning to those in Argentina and the rest of America in general, and good afternoon for those in Europe and Israel. I'm Marta Antonelli, and I will be chairing our second monthly virtual conference organized by the Argentinian Society for Neuroscience. It's a real pleasure and an honor for me to introduce Dr. Hermona Soreq today. It is difficult to summarize Dr. Soreq's extended career, but in a few words, I would like to tell you that Professor Hermona Soreq joined the Hebrew University in 1986, where she holds a Schlesinger Chair in Molecular Neuroscience. She's a founding member of the Edmund and Lily Safra Center for Brain Science. And interestingly enough, Dr. Soreq served as the first elected female dean of the Hebrew University's Faculty of Science from 2005 to 2008. She has authored more than 300 original publications, including 57 published in Science, Nature, PNAS, and Neuron and other high-impact journals, and more than 140 book chapters and reviews. She has an impressive age index of 86. Dr. Soreq combines advanced sequencing technologies with computational neuroscience and transgenic engineering tools to investigate microRNA functions in the healthy and diseased brain, pioneering research on acetylcholine-related processes. Her studies discovered cholinergic brain-to-body regulation of anxiety and inflammation, and found cholinomere silencers of multiple genes that compete with each other on suppressing anxiety and metabolic targets. She has an endless list of achievements, but to keep this introduction short, I will just stop here to let Dr. Soreq tell us her fascinating results. For those that might remember, she visited us in Argentina in 2001, and I just heard from her that she went to Argentina twice more times, when the ISN meeting was held in Buenos Aires, co-organized by the Argentinian Society for Neurochemistry at that time. She not only attended the meeting, but was one of the lecturers at the school held in La Estancia La Candelaria in Lobos. We are, of course, much honored to collaborate with her in the project we are developing in Germany together with Dr. Silvia Lobmeyer and Martin Frasch. Before we start, I would like to remind the participants that we will keep the microphones muted during the conference, and at the end, we will have plenty of time for questions. You will have two options to pose your questions. You can write the question either in Spanish or in English in the chat addressed to the host, and we will read it to Dr. Soreq, or you can ask to be unmuted and ask the question yourself. Please do not raise hands with the icon, since we will probably not see it. Dr. Soreq, thank you very, very much for accepting this invitation to our monthly conference. And in the name of the Argentinian for Neuroscience Council, we welcome you heartily. Thank you.
Thank you very much for this invitation. It's a real pleasure for me and a new experience to give a seminar that far away while sitting here in Jerusalem. And indeed, I would like to tell you about the non-coding RNAs and the cholinergic pathway in health and brain disease. And almost everything I've done in my life, in my research, relates to the cholinergic system. We believe that it is extremely important as regulators. As regulators of cognition, but also motor functions, and that microRNAs, and here is a model of one microRNA that binds acetylcholinesterase, serve an important role in making this happen. In a way, we are talking about small RNAs that control the big genes and serve as policemen, if you like. One new topic that we started about last year is the difference between men and women in the brain cholinergic system, and I'd like to talk to you a little bit about that. Okay, and a scientist is someone that knows more and more about less and less. And I've been teaching Introduction to Molecular Biology many years, and I have a confession. I told students 2% of our genome encode proteins, which do the work, and the rest is junk DNA. That was the word in textbooks. Today we know that most of the genome encodes non-coding RNAs, but that doesn't make them less important, possibly even more important than we ever understood. So I'm going to talk to you about the non-coding RNA, trying to simplify, as Leonardo da Vinci said, this is the ultimate sophistication. I'm not sure that we succeeded so far to simplify the complex message of non-coding RNAs. So here is the model of this microRNA. They all have numbers, no names. 132, which binds acetylcholinesterase and thereby stops its activity. So why is that important? For one thing, we find that a lot of the microRNAs in the human genome didn't exist there before the big primates evolved. So we are a relatively new species, and this is a new function that our genome acquired. So that's one. Second, we find that they control stress reactions. And stress responses also were subject to evolutionary change. We can think about the wild man in the desert being stressed because there's a lion running after him. We get stressed when someone we don't like says things that scare us. But what do? We go to the fridge and look for something nice as if a lion is running after us. So we kept the same machinery, but we react to different signals. Importantly, in the blood, what acetylcholine does is to block inflammation. So in the brain, it would block stress reactions. In the body, it might block inflammatory reactions. And I'd like to talk to you about that. This might change with age. Recent findings say that aged individuals subjected to long-term anticholinergic medications are at increased risk of dementia. Nobody knows yet why. So a lot of questions from babies to Alzheimer's disease, and let's start. So I would like to start with a story about Japan. In 1997, the Japanese TV showed one section of the Fukimon series, which millions, maybe hundreds of millions of kids were watching worldwide. Except that that session had 5 seconds of flashing lights. And that night, 800 Japanese children were hospitalized with acute seizures. So it took about 6 hours. That day, this particular movie was taken off TV. You can find it in YouTube, and it is very unpleasant to watch those flashing lights. But we were thinking, there are 3 questions that come up here. First, what is wrong with those 800 children? Why did they get sick? Second, what about the other 20 million Japanese children who also watched this? What protected their brain from getting epileptic? And third, what was the reason for this very rapid response? For a gene to be expressed, sent to the ribosomes, translated, the protein to take action takes much more than 6 or 8 hours. So then what we thought, and we in that case are Uriah Beckenstein, who was then a student in the lab, and Niba Mishra, a postdoc from India, they thought, could there be a microRNA which blocks certain genes in our brain and protects us from epilepsy? So the opposite job from what you would think, not stopping something and causing damage, but stopping something to cause protection. So how do we study that? Okay, sorry. Yeah. So we have a timeline, we have a system. Okay. And what we do is we design a mouse. I have a lot of clinicians in the family, and they say that I'm not a real doctor, I can only cure mice, which is true. So we engineered a mouse with excess of the microRNA that we suspected is involved, microRNA 211. And when the microRNA was reduced by antibiotic treatment, that mouse developed epilepsy. Then we went to human brains, and in Alzheimer's disease, we find that this microRNA is double in the level, probably in an effort to protect the Alzheimer brain from the seizures. You know, the 22% of Alzheimer patients have epilepsy that is practically untreated. So we looked at that in the brain of a lot of Alzheimer's patients, and we found that the cholinergic system in the Alzheimer brain controls lipid biochemistry, and most probably this is the pathway through which this microRNA protects the brain. So here is one example. What about another example? MicroRNA 132, the model of which I showed you, is blocking acetylcholinesterase. So what does this microRNA do? Here's a picture of cells that overexpressed it under stressful conditions. But this overexpression doesn't take place in the Alzheimer brain. A lot of the microRNAs are gone in Alzheimer brains that we see by sequencing, and microRNA 132 is declining more than others. You would say that makes perfect sense because this microRNA blocks acetylcholinesterase. So in the Alzheimer brain, we'll have too much of it. And that's very good that we are treating these patients with inhibitors of acetylcholinesterase, except that this doesn't block the inflammation, and the plasma cholinesterase activities actually correlate with the deposits of amyloid in the Alzheimer brain. And this is Mor Hanan, who was involved with this project and also completed her PhD very recently at the lab. So altogether, we decided to call those microRNAs that block the cholinergic pathway cholinomiers. We submitted an application to the ERC and won an advanced ERC support for looking at the complexity. Now, what does that mean? One microRNA can block many different messenger RNAs, and there would be some sharing between microRNAs, which is one way of complexity. Now, let's think about it in the context of the cholinergic system. Acetylcholine can block inflammation, and its levels increase in anxiety. But inflammation increases anxiety. Acetylcholinesterase degrades acetylcholine. So all of that can affect other targets of those microRNAs. In other words, during evolution, humans or big primates acquired a lot more regulation over the cholinergic system, which is of great interest for many research purposes and which Darwin would never have predicted, of course. So Bettina Nadorp came to our lab from Germany and did her PhD in the lab. And she said, let's accept that one microRNA can block many different targets. That means that one microRNA will block a pathway because a lot of genes in a certain pathway share some of their sequence motifs. So there should be microRNAs that block more than one gene in the cholinergic pathway. And she started looking for such microRNAs and found a lot of them. Here are 30, for example. 1/3 of them are primate specific. So what do they link with? They link with inflammation and anxiety, brain damage like stroke, which I'll talk about, cardiac disease, neurodegeneration, and pain. So far in the lab, we worked on 132 that I mentioned to you, 608, which I will talk about, and 125B, which I will talk about. So of the big project that we ran for 5 years, we so far managed to look in depth into only 3 of these cholinomiers. Okay. So what happened now? Okay. Okay. So we do have a change in with evolution, and we do have a selection of genes. How can we study what a microRNA does? So this is Iftach Shaked. He was a postdoc in the lab, and Shai Shoham, a staff scientist, and they created a transgenic mouse. So that mouse expresses excess of acetylcholinesterase, but we took off the region that interacts with microRNAs. So we have too much esterase, too little cholinergic signaling, and the microRNA can't do anything. What would that mouse look like? The first thing is these mice keep running. Okay, you see this is normal speed. They run so fast that they are 20% lighter in their body weight. So you would say, that's very good, they do sports activity. Except that if we put them in a maze where there is apple juice at the other side, control mice in the top find that juice very quickly, and the engineered mice, they don't find it. They go back and forth. They forget their keys. They don't know where they left their glasses. They're confused. They're making a lot of decision errors. And in addition to that, these mice show huge inflammation. So impaired cholinergic transmission because of insufficient quantities of a microRNA might cause disease. Would that reflect what happens in humans? So Shani Shenhar was a postdoc in the lab, and she's now a faculty member in Tel Aviv University. And she collaborated with clinicians who treat patients with Crohn's disease. So a lot of inflammation. In the acute cases, they operate to remove the inflamed intestine. And when we quantified, we found 10-fold higher levels of this microRNA in the human intestinal tissues under inflammation. So this phenomenon definitely reflects both rodents and humans. And then a clinician came to talk to me. This is Hanan Meidan. And today he treats corona patients, but when he came to talk to me, he ran an analysis of those microRNAs that share regulation of anxiety and metabolic disorders. And he came to show me the list, and I saw 132, so immediately I knew that this might be relevant. And I thought about the old cartoon. Remember the thin man and the fat man? The fat man is always upset and nervous, and the thin man is always happy. Could it be that there is a link between anxiety and fat accumulation? So how do you study that? You design a mouse. And that's what we did. In this case, we didn't engineer a mouse, but what we did was to feed them with a special animal diet that is 60% fat. And within 11 or 16 weeks, they doubled their body weight. The levels of microRNA 132 in their liver went sky high, and we could inject them with an antisense synthetic agent that blocks the microRNA. So now we have a system where we can study what does this microRNA do in a live context. And this was the work of Geula Hanin, who is now a Newton Fellow in Cambridge, a postdoctoral fellow. She engineered mice with excess 132, which gained fat. She fed the mice and injected the antisense. And here is the liver. What she does was to inject the tail vein with 3 mg per kilo of an antisense agent. And these are the photographs of the liver of these mice. You see that the untreated fattened mice have very big livers, very white, foie gras if you wish. But 1 week after injecting them with the antisense agent, the liver shrinks, becomes dark. We retrieve a normal looking liver without fat droplets. So that immediately reminds us the theory of James Neal from the last century, who said that we all carry genes that were profitable for our ancestors. Because you could think about the wild man in the desert getting stressed, running to a cave, and having to stay there for 2 months. People who could keep every bit of energy at those days had a benefit. So what we wanted to know is what happens, why is this microRNA so special? So today, Yonat Tzur is working on this project, and we looked at all of the steps of fat production, from glucose to cholesterol. We know the genes. We know the enzymes that are responsible for producing these compounds. We know the transcription factors that control those genes. And look at this, microRNA 132 blocks all of the transcription factors. No wonder it is so important. So I tried to block FOXO3 or P10 or P300. None of those had this effect, which led us to the conclusion that all microRNAs are equal, but some are more equal than others. Okay, what does Yonat do today? She's feeding human-derived hepatocytes with oleic acid, and she colors the accumulated fat and treats the cells with different antisense agents to find a better treatment that would be suitable for human disease. Okay, but you know what? Fattening is more frequent in women than men. And that's the case in many different diseases. So 2 years ago, there were 2 articles in Science, in one talking about heritability of disorders of the brain, like schizophrenia, bipolar disorder, and the other one talking about the molecular neuropathology, the transcriptomics. And I read these papers with great interest, and I was sort of disappointed to find that there was no reference to the difference between men and women. They had a lot of samples and they treated them all as equal. So I was so curious that I separated the data sets. They had to deposit the data sets. And the concept of these 2 papers was, 1, let's forget about mental disease, forget schizophrenia, forget bipolar disorder. We have a spectrum like the autistic spectrum. And when you separate the data to 2 data sets, the spectrum doesn't look that nice. So it wasn't a coincidence, it was on purpose. And therefore, I decided to look into these data sets, a lot of schizophrenia, a lot of bipolar cortical tissues, and find the difference in the brain. So this was a project shared by Geula Hanin, I told you about her, and Sebastian Lobentanzer from the University of Frankfurt. And they wanted to compare men to women data, run a meta-analysis, find a reduction of data which led us to the cholinergic system, design interactomes, and hopefully leading to application in therapeutics. And the paper was published in Cell Reports, and whoever wants it, just send me an email after the seminar, I'll send you the paper. Okay, so let's look at the comparison. We had a quadruple comparison from schizophrenia to bipolar and from men to women in both cases. When we looked at the genes that are enriched, look at this. The schizophrenic men showed completely different enrichment than women. And the enrichment led to response to organophosphates. Now we know what that means. Thanks to the recent Russian poisoning, we are remembering that organophosphate poisons block acetylcholinesterase. They induce a hypercholinergic excitation, and which may lead to death and severe disease. And I heard from a friend that at the Charité today, they have extreme security instructions to protect the life of this poisoned person. So that brings us to the human brain. So where are the cholinergic pathways in the brain? We all remember Rita Levi-Montalcini. She was remembered because of her discovery of nerve growth factor. And nerve growth factor therapeutics was tried for Alzheimer's, but didn't work, except that the cholinergic neurons in the human brain have different pathways, different roots, different type of neurons. And what Rita Levi-Montalcini found was the deep cholinergic nuclei, whereas we looked at the cortical cholinergic neurons. So we said, okay, we need to look at the sequences expressed in cholinergic cortical neurons in the human and the mouse brain. Luckily, there are a lot of data sets now of single cells, and we sought those cells that produce acetylcholine, that express cholineacetyltransferase. We found them. They also express the receptors. We recognize these cells. They share a lot of microRNAs, 1/3 of which are primate specific. They share a lot of transcription factors. No NGF. This is a different type cell. The cholinergic interneurons in the mammalian brain are not the same as the deep cholinergic neurons in the deep brain nuclei. So we looked, what do they react to? And instead of reacting to NGF, these cells react to inflammatory cytokines, interleukin-6, and the receptor is GP130. And that creates a response which would react immediately to inflammatory signals, and which highlighted a new microRNA, 125B, that Geula could show binds to acetylcholinesterase as effectively as 132. So here is one more microRNA that is more equal than others. Which brings me to the female-male question. I have a confession to make. I've been working with cell cultures for decades. I never think about the cell lines that I'm using in terms of male or female. We all know that HeLa cells come from a woman because her personal story is famous. But do you know about the other cell lines in your labs, what they come from? We decided to try to find closely related cell types that are cholinergic of male and female origin. And we found 2 such neuroblastoma cell lines, and we looked at the microRNAs they express. You may recognize this picture from the background behind my back. Apparently, there are microRNAs that are female specific, the pink ones, of course. There are male specific, the blue ones, of course. There are similar ones, and there are ones that are opposite. So we have families of microRNAs, and cells of male and female origin might show differences in their gene expression pattern as neurons. So this is the experiment we've done. We got these 2 cell lines. LAN2 is female and LAN5 is male, both from neuroblastoma. This is from a girl of 4 years, and this is from a boy of 3 years of age. They react to CNTF, which is a neurokine, and they develop processes. We are watching them in the lab. They increase the expression of cholineacetyltransferase, and they express microRNAs, some of which are equal and others are shared or opposite. Those genes that are blocked by them, again, show association with schizophrenia and bipolar disorder. So the cholinergic interneurons in the human cortex are important regulators of mental disease. And we could show that. If we differentiate the female cells, you see that all of the targets that are changed are female specific. If we mark, I hope that you can see the little blue dots are male cells, the little pink dots are female, and the purple ones are shared. And you can see that we have found cells that, genes that are specific to schizophrenia, mainly male, and males develop schizophrenia 10 years earlier than females and suffer more symptom severity and are all addicted to smoking, which says cholinergic. Some other genes are bipolar, mainly female. Females with bipolar disorder have much more severe symptoms than men. And there are genes that are shared. So I don't disagree with the spectrum, but I say there are two spectra. And when one tests therapeutics, you need to do that separately in males and females. Today, the situation is that all new drugs, and you know that as far as I do, are first tested in male mice, and only when they get to the clinical trials are they tested in human male and female patients. So half of humanity is being treated with drugs that were developed to benefit the other half. And we need to change that situation. Okay. So I was invited recently to write a review on this issue because the editor of Cell Reports made our data a resource, so it's shared. And we find that indeed there are huge pharmaceutical implications of sex-related RNA changes in psychiatric disorders. And this review will come out. They told me the proofs will come in a couple of weeks. And as we chatted before, this year we had a lot of time, so we used it to write more papers. Okay. So we need to remember that we live in a changing world. In 1950, the biggest community of human beings was younger than 30 years of age. Today, the biggest part of humanity is aged. And this fraction of humanity increases. These trees are the data sets from the UN. And we find a lot of articles telling us that cumulative use of strong anticholinergic agents are associated with dementia. And this was done in the United States and in Europe and in Britain. So it is a big data study, but nobody knows why that happens. So in that aspect, we decided to look at the age stage. So we selected another disease, ischemic stroke. Women suffer more from stroke than men, significantly so. And post-stroke processes first involve an inflammatory phase and then anti-inflammatory phase. So we thought this must involve cholinergic changes. Okay, so what would that changes look like? It could be those microRNAs that we found developed with evolution. But recently, there's a new family of regulatory RNAs that I'd like to tell you about. And this is a new story in the lab, yet unpublished, but it was well received at PNAS, and we are about to send a revised version to the journal. So this family of molecules involve fragments of transfer RNA. The most abundant small RNA in every cell because it's needed to enable translation. This is the project of Kasia Winek, a postdoc in the lab who came from the Charité and is doing that study. And it all started when we ran small RNA sequencing of blood cell samples from stroke patients. And we got the microRNAs, and then we got some sequences which people in the lab said, this is junk. These are fragments that are somewhat too long, about 30 nucleotides. They're not real microRNAs, but they did have sequence motifs that seemed to me reminiscent. And I need to tell you here that for my PhD project, I was requested to synthesize the left bottom quarter of tyrosyl tRNA. And I recognized the motif. And I said, this is not junk, these are small RNAs. Now, the next question we had is, so how do we validate that this is for real? If you run a qPCR amplification on fragment of a tRNA, you will see both the fragment and the tRNA, but that's not what we need. So we run acrylamide urea gels, we extract the small fragments, and we run qPCR and compare it to the sequencing data. And yes, indeed, there are small fragments of transfer RNA that increase after stroke in blood cells from patients. So remember, that's what transfer RNAs do. They are involved in controlling synthesis of proteins. But recent studies show that transfer RNA fragments might be essential for sperm maturation, for ribosome biogenesis, for angiogenin-induced translational inhibition. But angiogenin is increasing in stroke patients' blood, and nobody knew why. And I said, aha, this is our new story. Maybe this family of small RNAs is doing an important job. How do we study that? First, we ran a PCA analysis. Would control healthy people show different pictures of small RNAs from stroke patients? And they do. Second, we segregated the transfer RNA fragments. If it is a coincidence, we should get a mess. But you see here, it's not a coincidence. A lot of them came from alanine tRNA, which is different from in its function from arginine RNA and other tRNAs. So both microRNAs and transfer RNA fragments can function as blockers of other coding genes. So now we said, okay, let's look at the cholinergic ones. Here are volcano plots. Whatever is seen here is an increase. Whatever is seen here is a decrease. Now look at the red dots. These are fragments of transfer RNAs that target cholinergic genes. They go up. And these are microRNAs that target cholinergic genes. They go down. This is not a coincidence. This is what I call a change of guards. So could it be that microRNAs are good enough to handle matters under healthy conditions? But if an acute situation comes by, like stroke, then you need something that would be readily available, would not require any promoter to be synthesized, would be existent in any cell. That's transfer RNA fragments. So we discovered a new concept in the RNA regulation of the cholinergic system. Where do these transfer RNA come from? They find, we looked at studies that showed the collection of genes expressed in different subtypes of blood cells, and we found that CD14 monocytes are the origin of these transfer RNA fragments. Sebastian Lobentanzer also looked at the coding genes that are expressed in CD14 monocytes, and you see here that they express all of the cholinergic receptors, and both nicotinic and muscarinic, and the CoQ linkage protein. So we found a cell type that is in charge of controlling cholinergic activities in the human blood. Can we compare the targets of the microRNAs and the transfer RNA fragments that are changed in the post-stroke patients? If we look at the transcription factors, you see here, they're almost totally different. Not only these are different small RNAs, they control different transcription factors. And those transcription factors control the proliferation of CD14 monocytes. So if we are looking for the cell type in the blood that is responsible for the switch after stroke between inflammation and inflammation blockade, these are CD14 monocytes. So we submitted the paper to PNAS. The reviewers justifiably said, show us that these transfer RNA fragments can do something. So we took macrophages from mouse, we exposed them to LPS with or without dexamethasone, which is so popular these days. And indeed, the transfer RNA fragments we saw in the human blood go up under inflammation, go down under dexamethasone. What about human cells? If we take macrophages from healthy human volunteers and expose them again to LPS with or without nicotine, now we are looking specifically at the cholinergic system. Nicotine reduces the response, so this is a job of the cholinergic system. LPS increases it. And in one case, we even looked at the target. There is one TRF that was considered by mistake to be a microRNA, and people found its target, and we could show that that target is modified when we control this TRF by mimics or by antisense agents. We ran sequencing, we ran qPCR validations, and we now know that it works. Okay. Okay, sorry about that. So, last but not least, what about the DNA? The DNA is responsible for binding the microRNAs. And we know that blocking cholinergic system, like by insecticides, induces excitation in the frontal lobe, downregulation in the deep nuclei, and we know microRNAs that bind the acetylcholinesterase gene and might be responsible. We found a single nucleotide polymorphism that binds one more microRNA, microRNA 608, in acetylcholinesterase. So what will happen to carriers of this polymorphism? The microRNA cannot do its work. So we'll get too much acetylcholinesterase. True, carriers have 40% too much esterase in their blood. When my neurology friends tell me that some patients do not respond well to anticholinesterase inhibitors, this probably is why. 17% of our population carry this polymorphism. But the same microRNA has other targets, and those might be over-affected. So actually what we would see is a domino reaction. So the DNA is not less important than the small and long microRNAs. What happens in these carriers? So that was the other part of Geula Hanin's project. We looked at the SNP carriers, and they showed excess anxiety, excess acetylcholinesterase. One change in a non-coding region of the gene, they showed elevated blood pressure. Too much esterase in their blood, too much esterase in their brain. And interestingly, this SNP is highly abundant in African populations. 34%. 4% in Caucasians, and in the Middle East, Jews or Muslims, 17%. So if you ever doubted the theory of population migration from Africa to the Middle East to the West, here is another proof. And it could be, we thought, that this SNP was helpful. Maybe blood pressure was higher, anxiety was higher, but perhaps reactions to stressful situations were higher. Maybe that helped the wild man in the desert to survive. So how do we approach that? Alon Simchovich is an MD PhD student about to complete his PhD, and he ran a study together with Talma Hendler from Tel Aviv University, who's a psychiatric expert. We recruited soldiers and 69 Israeli Defense Force volunteers, and out of those, we genotyped them, 13 were carriers of this SNP. Now, we asked the question, would they suffer post-traumatic stress disorder? If they would, we would expect higher amygdala activity and lower prefrontal activity to stressful situations. We exposed them to part of the movie Silence of the Lambs, which is really unpleasant. And indeed, carriers showed higher amygdala activity. But when we compared their prefrontal activity, it was even higher. And it blocked the amygdala response. So they are more alert, but they do not suffer post-traumatic stress. And one of the reviewers in the journal actually had said, maybe they volunteer to become paramedics because they like the excitement, they react faster. Could be. So here is one more microRNA that is more equal than others. What does this do for post-traumatic stress disorder? We have in Israel 300 people who returned from captivity in Egypt or Syria during the 73 war. So they are all aged by now. They've been treated and followed by the army who takes care of them. And we had 99 volunteers who provided their samples. And out of those, 74 indeed are PTSD patients. The surprising thing is that 24 of them are not traumatized. So they came back after 2 years in the Syrian jail. This is really traumatizing, and they are healthy. So what we find is that those people carry a dual incidence of this SNP. It seems to protect them, and it's statistically significant. And this is a paper we are writing these days. Those that do have PTSD showed in their plasma exosomes, they showed microRNA 37D, which blocks inflammation. So again, all microRNAs are equal, but some are more equal than others. And a Chinese study found that carriers of a SNP in the microRNA itself are protected from inflammation. So now we are looking at both these variations. Last but not least, what do we do next? So this is Nadav Yayon, and he developed an algorithm that enables us to map and find the structure of each cholinergic neuron in a mouse brain. We have mice where all the cholinergic system is fluorescently labeled. And what Nadav did was to look at these cells after whisker plucking, which is stressful, and he found changes in genes that are known to control dendrite formation, which again is a paper under submission. That's what I came to tell you about. And I need to thank our students. You know, I looked at our students recently and found that at least 4 of them are graduates of the IDF intelligence service. So they came to the lab ready to do data analysis. I need to thank the patient volunteers who enabled this research, the colleagues in Israel and elsewhere who collaborated, and the foundations that enabled our support. So I told you about the cholinergic system, microRNAs, men to women differences, aging-related differences. This is my youngest granddaughter. Population implications and DNA relevance. And I'd like to end by citing Toni Morrison, who says that there is really nothing more to say except why. But since why is so difficult, one must take refuge in how. And this is what we do. Thank you very, very much for listening. And the lecture is open to questions.
Thank you very much. Thank you very, very much. And just imagine that you're receiving an applause from all the people. So thank you very much. And it will be very interesting. And I would like to invite people to make their questions either by chat or directly. So just let me know if you have any question.
Whatever. Yeah.
I think Sylvia Lagmeyer, I think I saw something. Is that so? Let me see.
Marta, do you need something?
Martin. Martin, do we unmute Marcela? Should we unmute him or he?
Yeah.
Can you unmute Martin?
Yeah, good.
Okay.
Hi, Martin.
Hi, Martin. Good seeing you.
I'll try to cut to the chase since probably people are on the clock. I enjoyed it so much. You know, so many ideas. Perhaps if I may ask just a general question first.
Please.
Do you feel that this extraordinary journey of discovery that you've been on for such a long time with trying to map the microRNAs, do you feel like you're you can say that you discovered instead of all the participants of the animal farm? I'm trying to build on the analogy of being more than equal here.
Yeah, I
do you feel like you
that.
have a map, or do you feel like despite all the particular examples you've shown, there is still a lot more in this map to discover?
No doubt.
Journey stands.
Yeah, there are thousands of microRNAs, thousands of transfer RNA fragments. There's work here for all of us for many years to come. And I think that what we have seen this last year is a revolution in that the entire world understands that RNA biology can become real therapeutics. So for years, I would submit patent applications and hear that I precede my time, that this is not yet relevant. But now it is. So I think if it ever was relevant, this is the time.
And I have another question, but I'll let you decide if I may ask it. I'm sure there is other folks.
No, it's okay. Go ahead, Martin.
You probably anticipate this, Mona, that I'm curious about two things, and we may not have time for both of them. And the keywords are the glia cells and electrical stimulation. And so I'm curious whether you have a sense of you know, what we can expect with regard, because you've already shown the important role of the monocytes in the periphery.
Yeah.
So I guess it's logical to expect that perhaps we have more to discover about neuroinflammation and the microRNAs. And the other, yeah, and then the question of electrical stimulation and whether that is a legitimate approach to therapy.
I let the science lead me. We went into CD4 cells because we found that they are relevant. So far, we found the neuronal gene expression pattern to be relevant, but I'm sure that there are processes that are handled by glia. So as soon as we find those, we'll move into that.
Okay.
Thank you.
Thank
It's
you.
nice seeing you after we exchange so many emails.
Yeah, and it's a pleasure. I could continue asking you for an hour, but I think that would be horribly inappropriate. So perhaps
Martin,
we'll
there
have
is a
another chance soon.
There's a question, Mona, from Diana Jerusalinski.
Okay.
Is there evidence that some microRNAs depends on activity? I don't know if it says activity.
that's it, I mean.
Diana, would you like to ask question directly?
But sorry, but I couldn't unmute before. On neural activity.
Yeah. Yes.
Coming. Yeah.
Yes. 132
Well,
is
perfect.
definitely.
Is great. Your talk was great. I'm thinking.
Thank you. Thank you. So microRNA 132 is neuronal, and it affects the circadian regulation of gene expression, which again is controlled by the cholinergic pathway. So yes, for this one, we know it's neuronal. 211 is also neuronal, and it controls inflammation in neurons. Yeah.
Thanks very much.
There's a question from Annalise Mojano saying, fantastic talk. Are microRNAs more efficient than tRNA halves to bind their targets?
This is the $64,000 question. We don't know the answer yet. This is very new. So our first paper on TRFs, I hope, will be now accepted to PNAS, where we'll submit it hopefully tomorrow. But the reviewers were very positive, and we did answer their questions. So I look forward to that. And we are proceeding to compare the activities in other systems. Yeah, it's really exciting because we have dual families and they might not do the same job.
Mm-hmm.
So Mona, in the same sense that Anna, the mechanism is similar to microRNAs or for tRNA. Do you think that they bind and promote the message, the decay or something like that?
So that's again an excellent question. The transfer RNA fragments are probably involved in many more functions via binding other targets, binding proteins, binding other RNAs. As you saw in the papers that were already published, they are essential for ribosome biogenesis, for sperm development. So it's a brave new world. We are, that's why we are so excited about them.
Yeah, okay, thank you.
Here I have a question from Lionel Muller-Igas. He says, great to see you, Mona, again. We met in 2001 in Argentina. Do you think that tRNA fragments regulation is only involved in acute situations, as you showed for trauma, or they might be involved in chronic long-term conditions, including neurodegenerative diseases?
The answer is the latter. We are already looking for transfer RNA fragments in the Alzheimer's brain, and we believe that they might be involved in the disease process, but that's too early. We need to validate that before I can talk about it.
Lionel, would you like to ask something else?
No, thank you. Just to say hi. Thank you.
Okay. Okay.
Great to talk to you guys.
And Martin Frasch says, connection of microRNAs to metabolic control and optimization.
Yes, we are looking into that. And we do have some targets, both of microRNAs and of transfer RNA fragments. Again, this is work in process. and the lab is working in half capacity these days, so it might take a couple months more.
Mm-hmm. Okay.
I guess, you know, this is a fascinating thought for me from an evolutionary biology perspective, asking, you know, is there something like a single optimization target that you can try to look at evolutionary optimization? Like what is it? Is it reduction of stress? Can reduction of stress dealing with avoidance of, you know, the coping with increased exposure to stress, can this be translated biologically?
Yeah,
into
yeah, we are involved in that. We use our small cohort as a kernel around which we are trying to expand into healthy human beings, hoping to find the answer to that. Yeah, you and I always think about the same questions, I know.
Here I have a question from Liliana Cancela. Liliana, would you like to ask it by yourself or just read it?
Well, I would. It was a fantastic talk. Do you hear me?
Yeah, absolutely.
Yeah. Okay. It was a great talk. I enjoyed it a lot. You are very clear to show all results, all findings. I was wondering about the role of this TRF MRI on acetylcholine, on the protective influence on inflammation, and on the other side, the protective influence of anxiety. How can you balance this kind of benefits and not benefit effects of acetylcholine from the point of view of clinics?
Yeah.
I was wondering this.
Yeah, well, here I must say that I completely failed in reaching the wish of Leonardo da Vinci to achieve simplicity. Rather, we found complexity. So, true, the same network might be responsible both for controlling anxiety and inflammation. And the balance between that requires small RNA controllers. But to say this is one, I can't. I had a big fight on the gut paper because we said this is a complex process. And the editor likes to find one gene and say, this gene is responsible to that feature. Life is not that simple.
It's a kind of balance, perhaps.
Yes, it is.
It's
A balance between networks. And every time we look into things, we find a new network, which is really exciting, but doesn't make things simpler.
Okay, and have you looked for another change on microglia, for example? Because you found some very nice results on monocytes.
Yeah, we are collaborating with Michael Hennecke from Bonn in a project looking for that. Yes, but again, this is a work in progress.
Okay.
Yeah.
Because it was a kind of interplay.
Absolutely. These are the immune cells of the brain, yes.
Okay, thank you very much.
You're very, very welcome. You were a wonderful audience. You ask all of the correct questions, and I apologize for not having the answer.
No.
Okay, it's okay.
Mona, I would like to ask you something. And since you were talking about schizophrenia and bipolar disorder, I was wondering what is the relationship of these microRNAs that target acetylcholine system? How do they challenge or affect the dopaminergic?
Yeah. So yeah, we are looking into the comparison between the cholinergic and the dopaminergic pathways. We find male-female differences there as well. And again, this is another new PhD student, and we are progressing. Yeah, we did publish this year the long RNA sequencing in embryo molecular medicine.
Okay. Okay.
Yeah.
Good. Yeah.
Yeah.
Okay.
Okay.
So I would like to know if there's more questions from the audience. It seems no. But so therefore I would like to thanks first of all, Mona, for your generosity in accepting our invitation. It was really a fantastic talk, very clear, very nice, even for people that are not in the field, so very nice. And I would like to thank all the participants also that